Hello and welcome back to the series of aquatic succession inside the house, where we see how an aquarium filled with just water on my windowsill slowly transforms into an entire ecosystem through various ecological succession steps, mainly by the introduction of organic matter through dead insects and the microbes they carry. For a more detailed explanation of ecological succession, you can watch the first video in this series. And to see what happened up until the start of the events in this fourth episode, you can watch the first 3 episodes. To very briefly recap, we started with only water, then dead insects fell in, bringing organic matter to the system. Bacteria and algae appeared, more dead insects fell in, the bacteria formed biofilms, the number of algae grew, more dead insects fell in, a lot more microbes appeared and grew in numbers, calcium carbonate crystals started to form, and eventually, after 3 months, the first living animals appeared in our ecosystem in the form of nematodes. What happened next? Well, let's find out. 3 months after the last events, a large addition of organic matter came into the system in the form of a ladybug. Sad for the insect, but good for the succession of our ecosystem. Apart from this beetle, a ton of tiny little flies fell in. My camera had a little trouble focusing, but pretty much every black speck on the water surface and on the bottom of the aquarium is a fly that fell in relatively recently. By now, compared to the last episode, the bottom of the aquarium was slowly getting gross, which is a good thing. Not only is there just a lot more stuff on the bottom in the form of all kinds of organic matter, but mostly dead insects, but by now there's also actual life on the bottom. The biofilms are getting really thick and there is some serious algae growth. The bottom glass pane is no longer clean and our ecosystem is going through an important step in aquatic succession. It's slowly forming a sediment layer. Just 4 days later, another huge insect fell in, again significantly contributing to the organic matter content. Here's my hand for scale. And here you can also see a little more clearly the other small insects and the bottom of our ecosystem, which is slowly coming alive. After half a year of succession, this aquarium that started off with just water is now housing a young ecosystem, which is slowly developing a substrate for potential future inhabitants. Cool! Almost a month later, this is what it looked like. The big insect was completely overgrown with algae, as is happening to anything that falls in at this point. The algae cover, specifically on the bottom of the ecosystem, has increased slightly. It's a subtle difference from a month ago, but a difference nonetheless. And on this scale, especially since it's happening inside, the ecological succession is a slow process. Now, this is going to be very difficult to see, but by now, some of the nematodes that appeared in this ecosystem 4 months ago at this point have grown big enough that they're barely visible with the naked eye, but really barely. This footage is shot with a macro lens and I also enlarge it digitally a bit. The interesting biofilm on the sides of the glass forming strings we saw earlier in the previous video had sort of disappeared in the meantime, but is now slowly growing back. I'm still not sure what's causing it to form strings like this. It almost looks like the biofilm has surface tension. If you look very closely, you can see a nematode just in front of the glass that is riding the current caused by me adding some water to the aquarium. This aquarium has quite a lot of scratches, which are the perfect brooding place for new biofilms and bacterial colonies. They also serve as nucleation sites for calcium carbonate crystals to form, which in turn potentially serve as suitable substrates for more microbial growth. Another 12 days later, a lot of the little flies that were previously on the water surface had now become stuck submerged to the size of the glass due to water evaporating and the aquarium being filled again. So every little black spot on the glass is serving as its own little microcosm, hosting many microbes that are feeding on the dead insect's body and harvesting its nutrients, allowing them to bring the organic matter from the relatively large insects into the microbial loop. Biology at work, baby! I figured this was a good time to, take a bit of a closer look. by very subtly grabbing our floating cover glass. The first thing we can see is a ciliate we have seen before, and which, like before, I still think is a spirotric, which have some very large cilia. These can help with swimming, just like the other cilia, but some species also use them to actually crawl around, acting like little feet. Pretty cool. In the last video, showing events 4 months before this, we looked at the very same cover glass under the microscope, and we could see that some calcium carbonate crystals had started to form on the glass. By now these crystals have grown considerably, and it looks like even more have appeared. It makes sense they would keep growing, as the concentration of calcium and other solutes in the water keeps increasing due to evaporating water being replenished with more tap water. Like I said in earlier videos, the tap water in Amsterdam is quite pure, very clean, biologically safe, there's no chlorine in it or anything, but not completely demineralized. Also, continued photosynthesis likely keeps increasing the pH, causing more calcium carbonate to come out of solution. These biochemical boulders are a playground for our nematode friends, which seem to be increasing in numbers as they're now not solely found in dead insects, but also roaming freely, on surfaces at least. Here in this shot we can see a nice collection of all different kinds of microbes. The bright green balls are some sort of algae cells, potentially the Chlamydomonas we saw earlier. The long diamond-shaped cells are a different kind of algae, diatoms, which have a glass exoskeleton. Very cool. The stick-looking thing in the center is actually a bacterial colony in the form of a chain, with a lot of bacterial cells next to each other. It's often cyanobacteria that do this, though I couldn't tell you if that's also the case here. Pretty much every little indiscriminate black or white thingy you see is an individual bacterium or archaeum. As you can see, many of them form these sort of aggregates, mini biofilms or the start of biofilms, bound together by extracellular compounds they secrete for this very purpose, forming colonies in a different way. And lastly, there is the elephant in the room, the very large thing swimming across the screen, screaming for attention. This is a ciliate, though not a spirotric like we saw earlier, because as you can see it doesn't have those very large cilia that almost look like little feet. I'm not sure what kind of ciliate it is, and they are a very large group, but I don't think I've seen it before, so it could very well be a new species for our ecosystem. Another nice example of succession, where more and more new species appear. When I say new, I mean relatively, of course, because the entire ecosystem is only 7 months old at this point, so not a single species has been here longer than that. So, as you can see, a lot can happen and exist in a very tiny part of a single drop of water, which is very moving in a way. It really puts things in perspective. Anyway, didn't mean to get all sentimental. Here, barely recognizable at this point, lies one of the cover glasses that tragically sank to the bottom half a year ago when I tried to let it float on the water surface. When it laid there on the bottom initially, it was really stuck on there. I always thought that was because of capillary action, but lately I've been thinking it might also have to do with Brownian motion. Maybe there's such a small amount of molecules between the two glass surfaces that there's almost something like a lower pressure due to Brownian motion between the plates than the pressure exerted externally due to Brownian motion from the surrounding water. Almost like an analogy to the Casimir effect on a larger scale. Whoa, easy tiger, you digress. Yeah, sorry, you're right. I'm not a physicist or a chemist. Anyway, the point is, it was really stuck. Then, around 4.5 months ago, some gas, probably oxygen, was forming under the glass. By now it's completely detached from the bottom, with a lot of stuff growing under it. Interesting, right? I sure hope so, but maybe I scared off everyone except the hardcore nerds. I want to see what lives on this cover glass, but I don't think I'll film the retrieval of this cover glass because it'll probably be a real pain. Haha, filmed it anyway. I can do whatever I want, you know. We have a microscopic view again. It took a while, sorry about that. So what we're looking at specifically here is the underside of the cover glass, the side facing the bottom of the aquarium. What we can see here are all the filaments that together are forming the greenish-brownish growth that was covering the cover glass on both sides, and which is also present throughout the entire aquarium, and especially on the bottom. Here we see the filaments at a much higher magnification. Determining what these filaments are made of is made difficult by the fact that even at this high magnification, no single cells are visible, meaning they must be very small, or these filaments aren't made up of cells. I'm not really sure what's happening here, whether they are bacterial, fungal, or something else, but statistically they are likely a chain colony of very small bacteria. But as you may have noticed, they're colorless. It's actually all the little green dots that are responsible for the color we see in all of this growth around the aquarium. Almost all of them are diatoms, of various species, so algae. Quite pretty, I think. The nematodes at this point seem to be so ubiquitous that they are even found under this cover glass on the bottom. Pretty neat. Here between these filaments we have something that looks like it might be an amoeba. Or might have been, I should say. I'm pretty sure it's dead. Sad. But such is life. Here, also among the filaments, we have another species of diatom, but interestingly, a species that we haven't seen anywhere else in the entire ecosystem. This could be a coincidence, of course, that is almost always a possibility, but it could also be that the little space between this cover glass and the bottom of the aquarium provides the ideal conditions for this species. Something interesting that you just might be able to see is that there's quite a lot of movement inside the cells, which I haven't really seen in diatoms before. Maybe they're completely infested with bacteria, or maybe it's supposed to be like this, but then I don't know what's going on. Interestingly, throughout the entire life of our little ecosystem, we've seen quite a lot of diatom species, but all of them are very small. Bigger species do exist, but not in here for some reason. Then again, the fact that there's any diatoms in here at all, when we started with just water, is pretty mind-blowing. Here where the cover glass was, you can see a really gross square surrounded by really clean glass. Neato! Time to return it to its not-so-natural habitat. And the one time I want it to sink, it lands perfectly on the water surface. How about that? Well, there she goes. I also wanted to take a little bit of a closer look at the large insect, because we saw cool stuff around dead stuff before. I only took a small part of this insect, of course, and here at the lowest magnification we can see that the microbial community around our deceased friend is quite colorful and quite active. Here we have a very nice example of a filament that's very clearly made up of a string of cells. In this case I believe they're algae though, and not bacteria. Totally tubular. There's various ciliates here, like this cute little one, and this one, which is one of those spirotrichs. There's a very interesting structure growing here, comprised of all the little black dots that are clumped together, as well as, once again, some form of filaments. Together they seem to be forming one large structure. These black dots really look like bacteria, and this could therefore be a bacterial colony or biofilm, but not like one I've ever seen before. Not that I'm an expert though. It looks like the filaments form structures which the bacteria grow around. Especially here you can see how it's almost like a tree, with the bacteria as leaves. Very interesting. Have I used the word interesting before in this video? I'm not sure. I'll try really radical. Maybe it's a biofilm made up of bacteria and fungal threads. How mysterious. Here we have a microbe with one long flagellum going totally crazy. And there's lots of algae cells and these spirotrichs. Here we have something spicy or spiny or prickly. A mean looking microbe with horns on top of its quote-unquote head. It's dead, but I think I know what it is, though I'll only tell you when we see a living one. Some of the microbe enthusiasts watching might have already spotted the way spinier microbe, which is also way smaller and harder to see. This, my friends, is a heliozoan, also known as a sun animalcule. It has an amoeba-like body, but with needle-like extrusions, made up of the cytoplasm of the cell, called pseudopods. They use these to move around and grab onto substrates, but mostly to hunt. Oh yes, these are fierce predators. In fact, you can actually sort of see it preying on bacteria as we speak. There's a lot of bacteria swimming around, but some are caught in the heliozoan's spines. Here we have what I believe to be a living relative of the dead and horned microbe we saw just before. It is a rotifer, and on the left side of your screen there's another nematode trying to steal the show. I've tried to identify it to a level more specific than just rotifer, but I failed to do so. What I can say, however, is that it's not a bedelloid rotifer. Those were actually the rotifers that appeared in the experiment with aquatic succession outside after 4.5 months. It's interesting and intriguing that it didn't take that much longer for rotifers to appear in the succession ecosystem inside, even though the aquatic succession inside is going much more slowly than outside. And also interesting and striking that it's a completely different species which never appeared in the outside ecosystem. It's also fascinating and really cool that we have these now, another new and big inhabitant. One could say a major microbe. I've seen a couple of them in here. I also wanted to take a little look-see-poo at this little patch of biofilm which was held up by some gas bubbles, presumably oxygen. There wasn't really anything we hadn't seen before, but there were a lot of nematodes here. And what there were even more of were diatoms. This nematode is swimming in a diatom soup. There seem to be about 3 or 4 different species. However, the majority of the diatoms, the grey ones, are dead for some reason. I wonder if we see a lot of dead ones here because they just sank to the bottom after they died. Or maybe there really is a bacterial infection going around and killing them all. Probably not though. Well, dear viewer, our ecosystem, which started out as just water, through ecological succession over 7 months, turned into the grave of a lot of insects and spiders, but more importantly, turned into the home of countless inhabitants. From free-living bacteria to bacterial colonies and communities forming intricate structures and biofilms, to many different kinds of algae in all shapes and, to a lesser extent, sizes, to larger protists like the ciliates, heliozoans and the fabulous rotifers, to actual animals in the form of our wiggly nematode friends. The number of all of them just keeps on increasing, with the nematodes for instance now being found throughout the aquarium, where before they were confined to dead insect carcasses. We can also see that the amount of life is increasing because of the baby sediment layer that is slowly starting to develop. All in all, this is a fascinating, engaging, exciting, absorbing, enthralling, engrossing, riveting and most of all, interesting journey, and I hope to see you in the next episode about aquatic ecological succession inside. That's it for now, goodbye. Mooi in focus, hé? Potverdikkie.